Hello everyone. Today we will discuss the staining process of some bacteria. There are mainly two types of staining process, the Gram stain process and the Zeden stain. The Zeden stain means Zeal-Nielsen stain. It is also known as acid-fast stain. Now first we will discuss about Gram stain. So in Gram stain there are mainly 4 to 5 steps for staining the bacteria. There are two types of bacteria, the gram-positive bacteria and the gram-negative bacteria. In gram-positive bacteria, the cell wall is made up of peptidoglycan, whereas in gram-negative bacteria, the cell wall is made up of lipopolysaccharide. So in first process, we will add crystal violet stain. It is a primary stain. We will add this crystal violet stain for 1 minute. After adding crystal violet stain, the gram-negative bacteria becomes purple in color and gram-positive bacteria also becomes purple in color. So the both types of bacteria becomes purple in color after addition of crystal violet stain. Now after 1 minute we will add a mordant. In mordant we will add iodine for 1 minute. So by which the gram-positive bacteria becomes purple and gram-negative bacteria is also in purple. Mordant we used so that the color will fix to the cell wall of bacteria. After addition of mordant, we will use a decolorizer so to differentiate the bacteria. We will add mainly acetone or alcohol for few seconds so that it decolorizes the color of gram-negative bacteria and they becomes colorless. So here the gram-positive bacteria is purple in color, but gram-negative bacteria becomes colorless. We have to do this process for few seconds only, because if we do this process for 1 to 2 minutes or more than 2 minutes, so the color of gram-positive bacteria will also becomes colorless. The bacteria becomes also colorless. So we will do this process for few seconds only. After this third step is most crucial step. This decolorizer step is most crucial step because we have to just remember that for how many seconds we have to decolorize this bacteria. Now the fourth step, we use this second stain to stain the gram-negative bacteria because they are becomes colorless. Now we have to stain them also. So we will use safranin red dye, which is a secondary stain, for 1 minute. So now the gram-positive bacteria are purple in color and gram-negative bacteria are red in color. So we can differentiate them easily. There are some organisms which are poorly gram stained. They cannot. It is very difficult to stain them in through gram stain technique. For example, mycoplasma. Mycoplasma is the smallest bacteria, so it is very difficult to stain mycoplasma through gram staining technique. The other examples like Rickettsia or Chlamydia. These two bacteria are mainly present intracellular, so the staining process is very difficult in them because they are intracellular, not extracellular. And other examples like spirochetes. Spirochetes are very thin and slender, so they do not impart color of gram stain. So these are also very difficult to stain through gram stain technique. Now after completing gram stain, we will move on to second stain technique, which is Zeden stain, also known as acid fast stain. It is known as acid fast stain because the bacteria is acid resistant. It does not impart its color by addition of acid, so it is acid resistant. So the name is acid fast stain. Now in Zeden stain process, the first step we use to add a carbonyl fusion. Now what is carbonyl fusion? Just break this word, carbonyl and fusion. The carbonyl is a phenol which is a mordant. Is a type of phenol which is a mordant, and fusion is a basic fusion which gives red color to the bacteria. The Zeden stain is in India. The Zeden stain mostly used for the bacteria like tuberculosis. So mainly we do the Zeden stain for tuberculosis in India. So in first step we will use carbonyl fusion. In second step we further heat it. the heating is act like a mordant, so that the color in the bacteria becomes fixed. So heating we will do for 5 minutes. Now after second process, the third as used like gram stain, we will add decolorizer. Here we do not add alcohol, we add acid with alcohol. Acid alcohol mixture we have to add, like sulfuric acid. After addition of sulfuric acid, we will add a secondary stain. The secondary stain like methylene blue. After addition of methylene blue, the background of the bacteria becomes blue in color, but the bacteria are red in color due to carbonyl fusion. The fusion gives red color to the bacteria. So the bacteria are red in color, but their background is blue in color, so that we can recognize them perfectly in the slide through microscope. So this is the all over process of staining the bacteria through gram stain and Zn stain. These two stains are very most common, and they are very helpful in the microbiology lab to detect the bacteria. I hope you understand. Thank you.